Hello, welcome to this web class. Hopefully you can hear me okay. Just give me a quick thumbs up or a smiley face emoji if you can hear me. Hopefully. There's a little bit of a lag, so it's always hard to tell. Oh, I need to mute my computer. Sorry, one sec. Okay. Yeah, how is everybody? Where are you guys joining from today? Got at least 3 of you. Anlib, Wow, am I saying that correctly? Mavita, hi Destiny, hi Hamza. Yeah, very good, thanks. Enjoyed the at least in, well, can't speak for the whole of the UK, but in London the weather's been really nice all week. It's been a good weekend. Oh, we've got everyone from all over. A few from England, Oxford, Qatar. I'm excited for the World Cup next year in Qatar. Thailand, nice. Wagwan. Good weather in Leicester. Nice. Yeah, I assumed it was kind of the whole of the UK. Yeah, I think we'll make a start. So today we're going through some A-level spec content. We're going to look at photosynthesis. So it's one of the first topics that most people cover in A-level. This combined with respiration. Although actually some people do learn about it later in the year. But yeah, hopefully you guys have gone through it before, and this would be sort of like a revision class. So let me just share my screen. I haven't done a YouTube class for about a month, and I can't remember exactly how to do it. Let me just check. Okay, that looks good. You can see a PowerPoint. Let me just swap that over. Like this. Perfect. So yeah, we're going to do photosynthesis. We're not going to do the whole of photosynthesis because it's a big topic. We're just going to do the light dependent reaction. So kind of like the first half. So yeah, we won't do anything on the Calvin cycle in this lesson. So a little bit about me. My name's Alex. I'm a biology teacher. I've been teaching for 4 or 5 years now, and I'm working with SnapRevise for about a year. And a bit about SnapRevise. So some of you might have already tried the website. If not, highly recommend it. Really useful revision tool. The format has slightly changed. So if you've seen the website before, have another look. The packages that we offer are slightly different now. But yeah, today, very exciting. We're giving away a 1-week free trial. So yeah, if you enjoy the class tonight and you think that's something I'd want to do more of and I found it helpful, then yeah, go onto our website, try it for a week, and yeah, see if you like it. Try before you buy, always good. So yeah, let's get started on the actual content. So the way we structure these web classes normally is before we start, we do a little bit of revision. This isn't a pyramid, and the questions are either topics that we've done in maybe the lesson before or 2 lessons ago to keep it fresh, or just things related to the topic, sort of like base knowledge that you need, some stuff you might maybe have from GCSE or from AS that you need for the lesson. And they generally they're tiered in terms of difficulty. So yeah, without further ado, first question, what is photosynthesis? How would you define photosynthesis? Seems like an easy question, maybe not as obvious as it first seems. And yeah, when I ask a question, just in case, yeah, you weren't aware of the format, it would be great. The more if you guys can all reply, basically, the more you engage with the class, the more you'll get out of it. So it's a good chance to have a go, and I'll try and look at as many answers as possible. It can get a bit tricky when there's loads of people, but I'll do my best. Okay, what have we got? Conversion of light energy into chemical energy. Nice. Using light energy to form organic products. Yeah, I like that. Yeah, these are good answers. Person asking, you're in year 12, is there any point watching this? Are you going to have to learn it next year? But to be honest, you'll find this quite hard. We're doing this sort of like as a revision pace rather than like the first time you've seen it. So you're very welcome to stay, but it might be a little bit challenging, but that's good to challenge yourself. So, but don't worry if you're finding it difficult to keep up, because this is like very fast to be going through it for the first time. Yeah, so production of sugars using light. That's just one definition. The ones I saw from you guys, they are also all good. In fact, some of them I would say are better. I kind of put a short one. So yeah, lots of nice answers there. Okay, so let's do these two together. So can I get the word equation and also the symbol equation for photosynthesis, or like chemical equation? So if you know the chemical one, maybe just do that. If you can't remember that one, just the word one, maybe let's do that one. Sky blue, yes, correct. Energy cannot be created or destroyed. Bringing a bit of physics into the lesson. Yeah, nice. So we have the word equation. Carbon dioxide. I rarely ever write that out. I always write CO2, it feels weird. Plus water goes to glucose, plus I can't even write oxygen. I'll go straight away for O2. Oxygen. So, and then as you guys have said, the chemical equation. Sorry, I don't know why people who have written the equation, for some reason it's being like, the comments aren't being allowed. It's saying, I'm not sure why. Maybe it's because, don't know. So we have 6 CO2 plus 6 H2O goes to C6H12O6 plus 6 O2. So hopefully this is something you learned at GCSE, because you do need it for GCSE, and it's still relevant the whole way through A-level. So it's also a nice one. You get two equations for the price of one, because if you just reverse this arrow, you've got the respiration equation. So really, really easy. Okay, so what happens to the products of photosynthesis? What could we say? Yeah, maybe it's like you said, too long of a word. I think it's like spam. The products, yeah, the oxygen is released to the air. So that's like what happens to the waste product. For plants, oxygen is a waste product, and then it can be used for respiration for plants and animals. So very handy that they do make excess oxygen. Otherwise, none of us would be here. Yeah, so the oxygen diffuses out the leaf. The glucose that they create glucose can be respired, or it can be converted into other molecules such as amino acids or lipids. So plants are producers, aka autotrophs. So they have to make their own biomolecules from scratch. So it's pretty neat that they can do that. So they can create amino acids, and they can create lipids all from the glucose they make in photosynthesis. Obviously, they need a few more things because glucose only contains carbon, hydrogen, and oxygen. So if you want to turn glucose into an amino acid, what else would you need? And if you want to turn glucose into a nucleotide, what would you need to do either or both of those? Yeah, for both, you're going to need a lot of nitrogen. So I don't know how I was going to write this. I'm just going to put it in brackets. Nitrogen. So you need a lot of nitrogen for nucleotides as well. And also, what's another thing you get a lot of in nucleotides? It's half of the sugar phosphate backbone. Yes, phosphorus. Nice. Oops, going the wrong way. Yeah, so they need a few other things, like maybe sulfur for some amino acids like cysteine and various other things, but that's the most important ones, nitrogen and phosphorus. So that's usually the main two ingredients in fertilizer, particularly nitrogen. So for plants to grow, they need to make protein. To make protein, they need nitrogen. Therefore, if you're a farmer and you want your plants to grow quickly, you give them extra nitrogen and then they will grow really fast. So useful. Okay, next one. Can you describe, so not just state, describe the organelle where photosynthesis takes place? For the fatty acids, they actually don't need any other nutrients or to create lipids in general. Lipids only have carbon, hydrogen, oxygen in. So you can directly turn glucose into a lipid. That one doesn't need any extra things. Yeah, so the organelle is the chloroplast. And there's various features of a chloroplast. Stroma is like the cytoplasm. Thylakoid is an internal membrane, or thylakoid membrane. And there's thylakoids and other things. I think we're going to look at them more on the next page anyway. So yeah, the light dependent takes place in the thylakoid membrane. Light independent takes place in the stroma. The light independent is a bit of a misleading name. It doesn't directly need sunlight to work, but it uses the products of the light dependent reaction. So if you don't have light, you also can't do the light independent reaction for very long. So yeah, a bit of a misnomer. Okay, final question. What is the structure and function of ATP? Yeah, nice, the name, adenosine triphosphate. Good. Yeah, so it's 3 phosphate groups bonded to a sugar, and then that's bonded to an adenine base. So phosphates, which sugar is it? That it's bonded to. It's a pentose sugar, which means 5 carbon sugar. But oh, Jess, you put the answer already. Ribose. Yeah, nice. Yeah, it's a ribose sugar. Therefore, it's based on an RNA nucleotide. If you get rid of these two, this is almost, there's a slight difference in the adenine. This is almost an RNA nucleotide. So that's where it originated from. And it's kind of the universal energy currency. Oh, sorry. Yeah, I forgot. The video covers it up, doesn't it? Yeah. Okay, there's a little bit. To be honest, you didn't really miss much. There's just another box and it's labeled adenine. So yeah. Anyway, I'm worried spending too long on these first questions. So let's push on. So these are the objectives. Look at photosynthesis and the different pigments, explain how ATP is produced, and then look at how NADPH is produced. These are the spec points for the 3 main specs. To be honest, it's pretty similar for all of them because it's a very integral part of biology, photosynthesis. So everybody learns about it. It's also in the international A-levels if you study those exam boards, or IB, or pretty much anything. If you're looking at biology, will be in there. Okay, so I'm just looking at the questions. I'll try to answer as many questions as possible, but we've got like 100 people in the class, so sometimes it's a bit tricky to answer everybody. So if I don't get to your question, don't think I'm ignoring you. I'm just trying to balance like getting through the lesson and answering questions. So symbol equation we already wrote. So actually, I'm just going to skip that to save some time. So what is chlorophyll? What could you guys tell me about chlorophyll? The lesson on the independent stage. We've got a lesson next week, actually, a YouTube one. I'm not sure what the topic is. I feel like it might be a different topic. If you wanted to go through, we've got all, if you join SnapRevise, we've got all the lessons, all the topics, all past recordings. So they're all yes, it's a green pigment. That absorbs light energy, something like that. Cool. So this is a chloroplast. We're looking a bit closer here. So this is the thylakoid. They're like discs, almost like a stack of pound coins. The membrane of those, the thylakoid membrane, is where all the enzymes for the light-dependent reaction are located. They're sort of membrane bound. This is a grana. And these are lamellae. Not everybody needs to know lamellae. I think it might just be OCR actually, but just in case, that's what they are. So you might notice there's a double membrane around a chloroplast. This is because chloroplasts were once, or we think anyway, chloroplasts were once free living organisms that got incorporated into eukaryotic cells. So there was some sort of like primitive bacteria-like thing that got enveloped, established a symbiotic relationship, and now over billions of years, it's so symbiotic that it's pretty much part of the cell. Yes, endosymbiosis. That's the theory. Nice, good knowledge. It's not in. I think it's in the IB spec, but it's not in all of the A-level ones. Cool. So chlorophyll is the best known of the plant pigments and also the most common, that hence why it's the best known. But there are some other pigments in the plant. So 2 of the most common ones are xanthophylls and carotenes. The color of these, well, actually, no, I'm going to get you guys to guess the colors based on the absorption spectrum. So I won't talk about those now. So why is it useful to have multiple types of pigment? What do you think? Yeah, nice, Ashani. So different wavelengths of light can be absorbed. Yeah, so this is called an absorption spectrum. This sort of graph thing here. On the bottom, we've got the wavelength of light. That's measured in nanometers. And then the absorption is just relative, so there's no real units there. But what you can see, so these are the colors are matched up with the wavelengths. So for example, 450 nanometers is like, well, sort of halfway between blue and green, like a bluey green color. So they can be a little bit confusing, but basically, if a color is absorbed a lot at that wavelength, you won't see much of that color. So it's where the lines are low, that's what color you see. So for chlorophyll, both chlorophyll A and chlorophyll B, they absorb quite a lot of the purpley, bluey light, and then they also absorb orangey red light. The colors that they don't absorb are green and yellow. Hence, they reflect greeny and yellowy colors back, and we see them as green, more green than yellow, but it does vary for the different plants. So yeah, if you've got low absorption, that means high reflection, and that's what we see, the reflected green light that isn't absorbed. So what do you think xanthophyll looks like? The colors. It absorbs a lot of green. Actually, to be honest, I'm not 100% if this line is that accurate, so maybe we won't do it because it is a sort of bluey color, and I wouldn't actually be able to guess that from the, yeah, it's actually bluey green, but it's not that obvious from the graph. Maybe that one's not drawn that well, but keratin is more accurate. So keratin absorbs blue, purple, green colors. Therefore, we see keratin as a orangey sort of red color, hence the name. It's also found in carrots, the pigment in there. So what else are we saying? Yeah, so these pigments are located together in photosystems, which or complexes that contain pigments which absorb light. So I think I saw somebody mentioned why they don't absorb all wavelengths of light, because that is kind of like the first thing I thought when I saw this when I was learning it at school. I was like, that seems weird. Like the more light you absorb, the better. So why wouldn't you want to absorb all wavelengths of light? Therefore, you could do maximum photosynthesis. The reason we think, and so then the leaves would basically be black. To absorb all light, you want to be black, a black color. That means you absorb everything. White color means you reflect everything. So yeah, why aren't leaves black? Therefore, they could do more photosynthesis. And the reason we think that is because if the leaves absorbed all the light, they would overheat and they'd struggle with like enzymes denaturing and too much evaporation of water. So it would actually be disadvantageous. Even though they would be able to absorb more light, there's enough other downsides to offset any potential gains. So not really relevant to A-level, but in case you were curious as to why that is. So let's just quickly define these words. What are 1, 2, and 3? ATP, NADP, and the thylakoid membrane. Actually, we kind of talked about ATP already, so I'm just going to put that one on. When hydrolyzed to ADP. NADPH then. I'm going to start writing this because aware, so got quite a lot to do. It's a coenzyme, which transports hydrogen plus electrons around the chloroplast. So a coenzyme is something that's needed for an enzyme to work. And these ones kind of work almost a bit like a Uber driver. They pick up H plus and electrons and they move them to different parts of the cell, specifically from the light dependent reaction into the light independent reaction, into the carbon cycle. Yeah, so they can act as both oxidizing and reducing agents, depending on whether they're oxidized or reduced. Nice. Cyclic membrane, where light dependent takes place. Okay, nice. So before we go through this, we should probably recap oxidation and reduction. So hopefully you guys have heard of these terms before, particularly if you do chemistry, you'll be very familiar. And you probably know the oil rig analogy. Annoyingly, this is written the wrong way around to do that. So oil rig is the way to remember. Oxidation is loss. Reduction is gain. And what are they losing and gaining? Oh, some of you guys have already put these on. Yeah. So gain of electrons, gain of hydrogens, loss of electrons, loss of hydrogens, H plus, or you can call these, also call these protons. I thought I had another word for them. No, just, yeah, protons. So if you hear the word proton, it's a H plus. Did we go through the carbon cycle already? No. We're not even doing the carbon cycle today. Don't worry. You haven't missed that. Okay, so how are you guys feeling with that so far? Hopefully happy. Give me a number or a smiley face. Or I guess just an emoji. The carbon cycle is the light independent. Yeah, exactly. Cool, nice, promising. Most of you guys seem pretty good. Bit of a mixture. People with 2s and 3s, is this the first time you've seen this? Or, because yeah, if it is, then I would not be surprised if it's a bit confusing. It would be quite hard to follow along for the first time. Okay, so let's do a past paper question. This is just quite a nice, simple 3-marker. We're just naming the parts here. So what are these? Nice. So inner membrane. B is stroma. Yes, it is. And C, a granum. Yeah. If you also put thylakoid membrane, that's fine. Because it's not exactly obvious which one it is. I would say probably granum more, but it could be pointing at the membrane. Okay, so let's go through the process. We're going to look at non-cyclic first. So what's the best way to do this? I think I'm going to walk you through it, just like drawing some arrows, and then I'm going to add the text afterwards. Yeah, that'll be the better way. So this is the thylakoid membrane. So we're now on the membrane. We've got these different things, different proteins, and well, yeah, let's just call them proteins, are embedded in the membrane. This one here is the electron transport chain. So similar to the electron transport chain you get in respiration. So let's start at the beginning. We get a photon of light that hits photosystem 2. So PS2, PS stands for photosystem, not PlayStation. What are they on now? Not really much of a gamer. Like PS2s were my like childhood. Well, we were just switching from PS1s to PS2s. I don't know what it's on to now. Is it like 4 or 5? This is A2. Don't know. Yeah, year 13 content. Okay, so light hits photosystem 2. This excites an electron. So we get an electron is excited and it moves to a higher energy state. That electron then moves through, I don't know, I'm doing wiggly line, that's the photon line. That moves through an electron transport chain, releasing energy, and it passes to photosystem 1. Then you get another photon of light, hits photosystem 1. This excites the electron to a higher energy state again, and it is then removed by binding to NADP, which when it gains the electron, it's reduced, and it also gains a hydrogen. So we call that NADPH. So how should I do that? NADP becomes NADPH. So yes, exactly. I see some of you guys have answered each other's questions, which is great. I love it when that happens. Exactly. That's the wavelengths that they absorb like best, or the strongest. That's the names. So the naming of these is a little bit arbitrary between the specs. Some just say like AQA. You can just call both of these chlorophyll. You don't have to say which photosystem it is, and you don't have to say which wavelength it is. Other specs, they want to see photosystem 1 and 2, or they want to see the wavelengths. So just have a look in your textbooks to just confirm what exactly you do need to know, because it's a bit different. Oh, up to PS5s. Okay, thanks. Well, someone said PS6. Have you got a new release? So let's put some labels on. So light hits chlorophyll. Chlorophyll in PS2. So how should we phrase this? PS2 is oxidized. Yes, oxidized. I always have to double think the redox reactions, which one's going which. It's losing an electron, oil rig, oxidation is lost. So PS2 is oxidized by light and loses electron, E minus. E minus is excited to high energy state and passes through electron, can't really write in the corner because the controls pop up. and passes through the electron transport chain. Next, moves to PS1 and is excited again by another photon of light. Cool. Nice. So hopefully that makes sense. That's unfortunately it's not the whole process. That's most of it. We just need to talk about what happens to the, well, basically we need to replace the electrons, and we need to talk about ATP synthase, which we haven't touched on yet. So yeah, hopefully that makes sense so far. I'm gonna have to go to the next page. So try and hold this in your brain so far. Because we're gonna, yeah, it's gone. I can go back if needs be. But yeah. So also occurring is a couple of other things. We've got a process called photolysis or photolysis, however you want to pronounce it, of water. Basically what that means is water is split apart using light energy. So it's kind of like the name makes sense. Photolysis. The word lysis means like breaking apart. And photo means light, doesn't mean a picture. So it's the breaking apart of water using light. So let's put that on. H2O is split into O2 plus E minus plus H plus. Now you can balance this two ways. I kind of prefer doing it this way, and then 4 and 4, because then you get one whole O2. Oxygen goes round as O2. So I think that's a nicer way of showing it. But some people prefer to do it like this. 1/2 O2, and then 2, and then 2. Both are absolutely correct. There's no real difference. But yeah, I'll leave that one on. So that's the photolysis of water. This creates electrons which can replace the ones that were lost. So to do non-cyclic photophosphorylation, we need water. If we don't have water, we can't do photolysis, we can't replace the electrons, photosynthesis stops. So water availability is one of the limiting factors of photosynthesis. We also see here why photosynthesis releases oxygen, because we're producing O2. So we're turning water into oxygen. So this is the waste product that we breathe. That bit doesn't really need to be added, but just checking up on any questions. Yes, H2O is indeed water. So let's add some labels to this one. Oops. So electrons are replaced by photolysis of water. Before I should probably explain it. So we've explained how the electrons are replaced. What is also happening is as the electron moves through the electron transport chain, it's moving from a high energy state or an excited state, you can call it either, to a lower energy state. as it passes through the electron transport chain. So it basically releases energy as it goes through. That energy isn't wasted, it's used to pump hydrogens across the membrane. So the energy pumps hydrogens in to the thylakoid. So that we're starting to establish a concentration gradient. So as that moves through, pumps in hydrogen, the energy released from the electrons pumps in the hydrogen, and we end up with a lot of hydrogen inside the thylakoid. So I'm going to draw them over here. We've got a concentration gradient. So high H plus conc, low H plus conc. So let's add that label. Movement of H plus through ETC, electron transport chain, pumps H plus across membrane. Oops, that's not how you spell across, whatever. That's still not how you spell across. I'm just going to leave it. Movement of H plus through electron transport chain pumps H plus across the membrane. This establishes an electrochemical gradient of H plus inside the thylakoid. Should we put that? No, let's put that, it's fine. So why do you think we call it an electrochemical gradient? What is that? Sort of like two words pushed together. Pooja, yeah, it's the movement of electrons through the electron transport chain that powers this. So we showed the electron doing that. This powers this, exactly. Powers the pumps. Yeah, so the electrochemical gradient basically means a gradient is a difference, or like a concentration gradient you'll be familiar with. So there's a chemical gradient. There's a difference in the abundance of the particles on one side, the protons on one side. There's more on this side than the other side. But because the protons are charged, there's also an electrical difference. So yeah, difference in charge, difference in abundance. So both of those combined are establishing an electrochemical gradient. Maybe that was a bit, I don't know, I'm worried I'm going into too much detail rather than just focusing on the core bits. Okay, electrochemical gradient of H plus. So H plus moves through ATP synthase. So the H plus, it's an ion. Hopefully you guys remember from AS, ions are charged. They can't move through a phospholipid bilayer. So ions can't get through the membrane. The only way they can even out the concentration gradient is to pass through ATP synthase. So that's what they do. They go through there. That releases some energy, and that energy is used to stick together. Sorry, I'm just gonna rub this out, I've run out of space. Oops. I really struggle if I try and write and speak at the same time, my brain isn't capable of doing both. ADP plus PI goes to ATP. So essentially, the energy from the concentration gradient is used to synthesize ATP. So we've made ATP. So let me add that label. moves through ATP synthase, turning ADP, adenosine diphosphate, plus inorganic phosphate into ATP. Final note, this is known as chemiosmosis. Or chemiosmotic theory. Oops. The chemiosmotic theory. Okay, so that was a lot to take in, I'm aware. Especially if that was the first time you've seen it, that will probably be extremely hard. But yeah, any specific questions that you guys have? I can't like help with I don't know anything, but if there's something specific, I could answer now. Otherwise, we'll move on. How is NADPH produced? Yes, good question. So the NADP is a coenzyme. It's just like present in the sort of in the chloroplast, and the NADPH, that's basically the NADP. Let's write an equation. Plus E minus. So this is just a coenzyme floating around. The E minus was from here, plus H plus. The H plus was what was coming through the ATP synthase. This is what makes the NADPH. So yeah, that's kind of like where it comes from. Could I explain cyclic? Yes, we're going to look at cyclic later. Photolysis, yeah, you could kind of say either. It's like, it's specifically the photosystem that actually causes it. So chlorophyll actually does the photolysis. So I'd probably say on the membrane is where that's happening. Yeah. Photoionization is the excitation of the electron. So we could call this so many lines. Maybe I shouldn't draw it. So photoionization, the excitation from a photon. Yeah, what else? Where do the protons come from? They come from photolysis. So yeah, that's creating protons, and then they pass through. It's not necessarily the same one. So water is split in photolysis, and then yeah, the electrons pass through. It's not the same hydrogen that's pumped necessarily. It's just like each time electrons move through, hydrogen is pumped across. But yeah, that's where they come from in general. No, photosynthesis definitely not in humans, which is a shame because it would be quite useful as a little energy boost. We'd have to then be green, which would be the downside, or an upside, depends how you look at it. But actually, it wouldn't actually work very well for humans. We have very high metabolic rates, very high energy demands, and actually a very low surface area. So to create enough energy for a human, we'd need a massive surface area. So we'd have to grow, basically we'd have to grow a lot of leaves. And like, I reckon if you had massive sort of like wings and they were all covered in leaves, you might have almost enough to sort of like keep you going if you didn't do very much. But yeah, it basically wouldn't work at all for humans. Okay, so what's supposed to happen on this one? Oh, it was just talking about the electrons. So electrons plus H plus. We've already talked about this. I just drew it all on the same slide. Makes the NADPH. So equals reduced NADP. NADP equals oxidized. Well, yeah, oxidized NADP. So this one has H plus E minus, no H plus E minus. Why are people talking about fungal diseases? Did I miss something? Okay. So I think that is most of what I wanted to look at for that. Yeah, hopefully that's making some sense. If you've gone through it before, it will be much easier. If it's the first time you're seeing it, it's quite a lot to take in. But let's do some questions on it. Oh, annoyingly, the first question, the answer is on the screen. So, and you can't even see the question. So, well, never mind that one. We'll go straight to the second one. You need, yeah, we're going to talk about cyclic next, where you don't actually need photosystem 2. But for non-cyclic, you need both photosystem 1 and photosystem 2 for that to occur. So describe how NADP is reduced in the light-dependent reaction of photosynthesis. It's worth 2 marks. So you need to give 2 statements. So reduced. Reduced, reduction is gain. That should be basically what you're thinking of for your answer. So how is NADP reduced? Basically, how is anything reduced? What happens? Yeah, so accepts electrons, accepts H plus, or gains. You could say either. Each of those is worth a mark. So actually quite easy to get two marks for that. So this one, oh no, I thought there was a question. So use the information in the diagram to explain the role of chlorophyll in photolysis. So what do we think? What's H plus? Is a hydrogen ion. Equals proton, or it's all synonyms for the same thing. So the role of chlorophyll in photolysis for 3 marks. So we can say it absorbs light energy. Loses an electron, or is oxidized. It's kind of the same thing. Therefore, accepts electron from water. Or you could say like acts as an oxidizing agent. And actually, chlorophyll is a very powerful oxidizing agent. Water is pretty stable. The oxygen and the hydrogens are pretty well bonded together, but the chlorophyll is able to split it apart. It's able to grab the electrons and split water. So it's a pretty powerful oxidizing agent. Yeah, these questions are quite tricky. Like knowing what to say is hard. These are definitely challenging questions. So next one, also worth 3 marks. How the energy of light is wait, that doesn't make sense. This is missing some of the. Oh, sorry, it's from this first bit. Okay, now I understand. So use the information diagram to show how the energy of light is converted into chemical energy in the light-dependent reaction. So how are we making chemical energy here? What form is the chemical energy in that we make in the light-dependent? In the light-independent, the chemical energy we make is glucose. But in this one, yeah, we've just created ATP. So produce ATP, that's worth a mark. And basically explain how it happens. So excited electrons pass through electron transport chain. That's a tick. Pumps H plus across membrane. That's also worth a mark. Electrochemical gradient. Pumps. Oh, sorry, not pumps. Forms ATP passing or by passing through. Sorry, I really ran out of space there. ATP synthase. Cool. So yeah, don't be like put off if you found those questions really difficult. They are really difficult questions. Yeah, if you were following along okay as we were going through the diagrams, then that's great. That's yeah, for a lot of other questions you would be able to answer easily. They were challenging ones. So these questions are from mixed exam boards. Yeah. Kind of aware of time. We haven't got, it's basically like been an hour now. I'll quickly talk about this. So we talked about non-cyclic. We're now going to look at cyclic. So what can happen is rather than the electrons passing to NADP and being lost and having to be replaced, they can just stay in the system. This is just in PS1. So basically, if we go back to our other diagram, it would be like this. It just goes up through the electron transport chain and then back. Each time it gets hit by light. Can't draw that there. Oh no. It's doing this subtitle thing. But it just stays in this loop. So that's cyclic. what that means is you don't need water, which is good, but you can't make glucose from it. All it makes is ATP, because each time it passes through, that's pumping hydrogen in, therefore we can make ATP. So no photolysis, only PS1 makes ATP, no NADPH made. Or should I say, no NADP is reduced. Okay, so yeah, that was very quick, but I've kind of run out of time, so I just wanted to just put that on there. That this is useful because it creates ATP, which can be used in the Calvin cycle. Because if you remember, the Calvin cycle requires ATP, so it's kind of like stockpiling for that. Okay, so we're gonna end there. There is a little bit more of the lesson, a couple more questions basically. We haven't got time to do them today. In a normal web class, what we do on the website, there's a few less people, so we usually get through a little bit faster. So we probably would have finished if that been a normal class within the hour. So yeah, hopefully you guys enjoyed that lesson and you learned something. If you enjoyed the class, then you've got a couple of options. You can set a reminder on YouTube for these classes. We do one per subject per month. So the biology one is actually next week. It just happens to be that's the schedule. So yeah, set a reminder for your classes. If you do physics and maths or chemistry as well, oh, there's no chemistry on there, then obviously feel free to attend the other subjects as well. Then, yeah, wanted to talk to you about our tutoring package. So there has been some slight changes on the website. We now run tutoring and the classes as a separate package to the other website content. So yeah, for the subscription, which is £49 per month, you get access to 4 web classes. We go through all of the topics, and you can access all of the past lessons that we've done. So you can come to the ones in the like the live sessions, but you can also look at all the backdated catalog. Yeah, and basically it's kind of like getting the benefit of tutoring because it's live and because you can ask questions and the tutor will answer your questions. You're getting all the benefits of one-to-one tutoring, but for a fraction of the price. So I do like one-to-one tutoring in London as well. And yeah, it's £50 an hour sort of minimum you're looking for like one-to-one tutoring. So attending these web classes where there's maybe like, sometimes there's only about 3 or 4 people in a class. So you're getting like really like a lot of attention, individual attention for an absolute bargain. So I think it's a really good offering. Let me quickly show you the website. Just a reminder, you get a week free trial. This is the code. And I'm going to quickly walk you through the website. So let me just move over. Is it this one? No. So this is our site. This is what it looks like. The other courses you'll find on here, and you can join those. They're different packages. What I was offering you today was this tutoring package. So if you click on this, follow the link. These are the subjects that we do it in at the moment. It's only science and maths for now, but we're going to hopefully add some other subjects. So let's look at biology. This is the page, some information about it. This is the package. And yeah, these are some free samples. We've also got ones on YouTube, like past sessions, like the one we did today. So you can check those out as well. Our classes are split into 2 different types. We've got web classes. The one we did today was a web class. It's basically like structured. We go through a topic, do past paper questions, and that's a web class. A drop-in session is whatever you guys want to work on. So if you've got questions that you want to go through, or a topic that you're not really that comfortable with and you want to go through, the drop-ins are your opportunity to ask questions, to select topics. So there's kind of like, yeah, two options. And I think there's not much more to show you. This is the schedule. So for example, this week, if you joined up, you could access all of these past recordings, and we've got recordings like from the last 12 months. And the schedule for like the rest of this month, where are we up to? So Topoka, the other biology tutor, she's doing a lesson this evening. And then I'm doing like 4 more next week. So actually we're doing 5 sessions a week at the moment. So even more bang for your buck if you sign up. So this week, what am I doing? Enzyme reactions with year 12, 2 drop-ins, and we're looking at AQA essay style questions so that one's actually, it's not normally only specific to one exam board. That's quite unusual that we're doing that, but it's really helpful for any AQA students. And yeah, that's Topoka. That's obviously me. I think that's everything. So yeah, again, thanks for joining. Thanks for giving up your Sunday evening to study. It shows that you're very dedicated, so that's good. And yeah, hopefully see you on one of these classes on YouTube in the future, or even better, in our like web classes on the website. Yeah, no, you're very welcome. Tropical flea. That is not very nice, but I guess it kind of is true. Yeah, you're only competing against each other, so yeah. Cool. Yeah, have a good evening. You're very welcome. Good. Glad to hear it, Hamda. Actually, yeah, Daniel, that's very true. The exams this year are internal, yeah. So yeah, I hadn't even thought about that. Just unnecessarily aggressive then. You've got an exam on this tomorrow. That is very good timing then. Okay. Yeah, I think I'll end it there. So yeah, thanks again. Enjoy, and see you soon, hopefully.